We would have never guessed what those are for. Well, it is the technology needed to make GMOs. But do we really know what GMOs are?
Hi, my name is Zineb. I'm going to talk about genetic engineering. Well, it's a modification that occurs in a cell's genome by adding a gene, which is a piece of DNA that codes for specific genetic information. At the end of this process, we get something that never existed before in nature. Isn't that miraculous? Well, let's discover why does it take so many years of research by going through its steps. First step is to find the gene of interest and isolate it. Well, to do that, we need restriction enzymes that will cut the DNA in precise points depending on the nitrogenous bases order. The other thing that we could do is using another enzyme that is called
reverse transcriptase.
It produces DNA from RNA messenger thanks to the complementarity of nitrogenous bases. Now that we have our gene, we can insert it into a plasmid. A plasmid is a circular DNA of bacteria. To insert the gene, we need another enzymes like ligase that will glue the gene into the plasmid. So to permit for the gene to be expressive and effective in this new piece of DNA, we need to add a promoter and a terminator gene. Those help when the gene wants to be expressive when we move from DNA into RNA messenger and into amino acids thanks to what we call a transcription. Now that we have our plasmids that are genetically modified, we can put them back into the bacteria. Well, some of them will accept this plasmid, some others will not. We need to distinguish now which of those bacteria are genetically modified and which are not. To do that, we can do what we call hybridization, which is adding a sequence of DNA that will make a bond with the gene that we added. So every radioactive bacteria is the one that we are looking for. We can do another thing, which is add a specific antibiotic, because every plasmid that are genetically modified has a resistance toward an antibiotic. So the bacteria that will survive this presence of these antibiotics will be the ones that we are looking for. Now we can let them reproduce and express themselves. So why is genetic engineering so revolutionary? Well, it's helped a lot in developing the industrial and agricultural field. It's also helped in the pharmaceutical field because now we can produce human insulin. And this is how we do it.
As we see in the picture, we take the gene responsible for producing insulin from a human cell, insert it in a plasmid. After the reproduction of bacteria, they can produce insulin the same way the human cell did. That's why we say DNA is universal.